So today we're going to be looking at assessment statement 11.1.2, which asks us to outline the principle of challenge and response, clonal selection, and memory cells as the basis of immunity. The first thing I want to do is just to remember the role of monoclonal cells, or monocytes, as we called them yesterday. These are a type of white blood cell that's going to ingest pathogens by phagocytosis, and then it will basically digest the pathogen but take the antigens from the pathogen and display them on its surface. So basically this monoclonal cell or monocyte is going to put on the disguise of the pathogen in order to alert the rest of the immune system about what type of pathogen has entered the system. Okay, so let's take another look at this assessment statement. It asks us to first outline the principle of challenge and response. And challenge and response is really pretty straightforward. It's basically the principle that says that our system is being challenged by a pathogen, and our system is going to respond with the selection of or the clonal selection and memory cells being developed. So what I'm going to do next is go through the whole process of clonal selection and the production of memory cells, which is diagrammed here. Basically what's going to happen first is the pathogen is going to enter the system, and so here we have the pathogen and it's entered the system. It's going to be reproducing most likely as it's entering, and at some point a monocyte or monoclonal cell will identify this pathogen as being something that is not a self cell. So remember that all of our own cells have self antigens. This pathogen will have foreign antigens. The monoclonal cell or monocyte will then ingest the pathogen by phagocytosis. And as you can see in the diagram, this monocyte also has a lysosome present, and that lysosome will be combined with the pathogen to break down the pathogen, rendering it ineffective. However, the antigens from the pathogen will then be displayed on the outside of the monocyte. So here we see the monocyte has put on the disguise of the pathogen. The monocyte will then travel to the lymph node. The lymph node is the site where lymphocytes are stored, and remember we have two types of lymphocytes, both B lymphocytes and T lymphocytes. And so what's going to happen is this huge number of lymphocytes that we have stored are going to be selected, and they're going to be selected based on the antigens that are present. So as you can see from the diagram, and this is simplified, but they're based on shape. So you see that there's a T cell that matches the green antigen and a B cell that matches the shape of the pink antigen. So this is the selection process, the clonal selection. These B and T cells are then going to be cloned. They're going to basically make an army of lymphocytes specific to this pathogen. In this case, we've got both plasma cells, B memory cells, and T killer cells being produced in mass. At this point, plasma cells, we'll discuss those first. Plasma cells are going to be the cells that are responsible for making the antibodies. Now, the antibodies are shape-specific or antigen-specific, and the antibodies will be produced by the B cells and then sent out into the system to mark the pathogens for destruction. Now, it's important to remember that antibodies don't actually cause destruction of the pathogen, but they mark it. And so as you see, these little yellow antibodies will go out and they will attach to the pink antigen on any pathogen they come in contact with. All right, once they've been marked, this is where the other cells come in. So now we're going to talk about T killer cells. Once the pathogen has been marked, that is a signal for the T killer cells to come in and destroy that pathogen. And so this is where the pathogen actually, some harm is done to it. In this example that I'm showing you, the T killer cell actually causes the pathogen to lyse or explode. And it does this by inserting a cylindrical protein into the cell, which causes the lysis. So basically what's happened is the cells have been marked by the antibodies, and then the T killer cells have gone out to destroy the existing pathogens. The last type of cell that we've talked about is the B memory cells. B memory cells, their role comes throughout the remaining life of the organism. And they will remain in the system throughout the life of the organism, and anytime a similar pathogen or the same pathogen enters the system, the B memory cells will act quickly. Basically, what the B memory cells allow is for the body to skip the first couple of steps. So we get to skip the monoclonal digestion here, we get to skip selection, we get to skip cloning. Those B memory cells will stick around in the body and be ready to respond quickly to any further attacks from this specific pathogen. The next assessment statement we need to consider is 6.3.6 and 11.1.4. So this is just a repeated assessment statement at both the standard and higher levels. And it just asks us to explain antibody production, which really we've already done, but I'll just go through it quickly again. So once B cells are selected, they're going to produce plasma cells through cloning. Those plasma cells will then produce antibodies. The antibodies will be circulated in the bloodstream, and antibodies will bind to the antigen that they were made for and mark the pathogen for destruction. Now, just on kind of an interesting side note, there's a few different types of destruction. The one that I described before, like I said, was lysis. But there are two other types that we'll talk quickly about. The first is agglutination. And agglutination is basically when a pathogen is marked by an antigen, that antigen will match up with other antigens that have been marked. And so it effectively makes a large clump of pathogens. So several pathogens are all bonded together by their antigens and antibodies. And this makes them more vulnerable to being engulfed by phagocytic leukocytes. And basically just makes the process more efficient, as well as multiple pathogens can be ingested at one time. So kind of interesting. And then the last type is neutralization. And the reason that pathogens are often dangerous to us is because they're producing some kind of toxic product. So something about them, either their metabolism or their waste, something that they're producing is toxic. And so neutralization is where an antigen will attach to the pathogen and will inhibit the production of this toxin. So we'll inhibit the pathogen from actually releasing whatever it is that it's producing. And then, like I said, the last is lysis, which is where the T killer will insert a cylindrical protein, causing the cell to lyse or explode. All right, please take a look at the textbook for more information and be ready to ask some questions in class tomorrow. Thanks.